Induced mutation. In 1927, Muller reported the induction of mutations in Drosophila by X-rays, and in 1928, L.J. Stadler obtained similar results with barley. It was soon discovered that other radiations such as UV light and also some chemicals can act as mutagens or mutagenic agents that produce mutation. The discovery of existence of large number of mutagens in our ordinary environment was and is continuing to be a matter of considerable public concern. The mutation has permitted the identification of numerous genes in all types of organisms. What are mutagens? Mutagens are of two types, physical mutagens and chemical mutagens. All forms of life are exposed to energy-rich radiations capable of causing chemical changes in DNA. When such a change escapes repair by cellular mechanism, a permanent heritable mutation results. The physical mutagen includes the radiation-induced mutations. That portion of electromagnetic spectrum containing wavelengths that are shorter and of higher energy than visible light can be subdivided into ionizing radiations and non-ionizing radiations. The ultraviolet and ionizing radiations are responsible for about 10% of all DNA damages caused by non-biological agents. Effects of UV light on DNA. UV light is the part of electromagnetic spectrum between 200 and 400 nanometer with shorter wavelengths than violet of the visible spectrum. This range is further divided into short wave 200 to 280 nanometer, that is UVC region, middle wave 280 to 350 nanometer UVB region, and long wave 315 to 400 nanometer UVA region. The shorter wavelength UVC light carries with it a lot more energy than its long wave UVA counterpart and is more damaging to DNA. UVB and UVC rays are of two types of high energy radiation that are capable of ionizing molecules in a process called photochemical reaction that leads to a new molecular product. And this slide shows the UV region of electromagnetic spectrum. The UV rays can reach the ground surface, that is the troposphere, and can cause ecological and also health impacts. Ultraviolet rays do not possess sufficient energy to induce ionizations. They are, however, readily absorbed by bases such as purines and pyrimidines, which then enter into a more reactive or excited state. Because of their lower energy, they penetrate tissues only slightly, usually only on the surface of the cells in multicellular organisms. The maximum absorption of UV rays by DNA is at wavelength of 254 nanometer. Maximum mutagenicity also occurs at 254 nanometer, suggesting that the UV-induced mutation process is mediated directly by the absorption of UV rays by purines and pyrimidines. The UV damage in DNA occurs via two distinct types of mutations, dimerizing mutations and oxidative mutations. Dimerizing mutations. The most common photochemical product in DNA is a cyclobutane pyrimidine dimer. This product forms when two adjacent pyrimidines become linked covalently by their CC double bonds. For example, the two thymine bases can link together or two cytosine bases can link together. These four carbons form a cyclic ring or cyclobutane that links the two pyrimidines, thus creating a chemical intermediate that is not normally found in DNA. This photochemical product causes a structural change in the DNA that prevents the pyrimidines from base pairing and prevents DNA replication. This slide shows the common type of DNA damage, that is when the wild DNA is exposed to UV light. The pyrimidines, especially the thymines, they can form a cyclobutane ring and a dimerization occurs. And we are getting a pyrimidine dimer. In vitro studies shows that the pyrimidines absorb strongly at 254 nanometer, and as a result they become very reactive. The two major products of UV absorption by pyrimidine appear to be pyrimidine dihydrates and pyrimidine dimers. Several lines of evidence indicate thymine dimerization is probably the major mutagenic effect of UV. Thymine dimers appear to cause mutation indirectly in two ways. These dimers apparently perturb the DNA double helix and interfere with the accurate DNA replication. And the occasional errors are made during the cell processes for the repair of damaged DNA, such as DNA containing thymine dimers. Next is about oxidative mutations. UV exposure doesn't always lead directly to mutations in the DNA. In fact, UVA radiation commonly causes the creation of a free radical that then interacts with and oxidizes DNA bases. These oxidized bases do not pair correctly during replication and causes mutation. One example of this is a guanine to thymine transversion mediated by reactive oxygen species. The oxidation of guanine into 8-oxoguanine prevents the hydrogen bonding required to base pair with the cytosine. That is the normal base pair for guanine is cytosine, but there is a change in the structure of guanine. It is converted to 8-oxoguanine and it cannot base pair with the cytosine. Instead, during replication, 8-oxoguanine can base pair with adenine only via two hydrogen bonds. When the second strand is synthesized, that is in the second replication, the base position originally occupied by a guanine is then replicated with a thymine, leading to a G to 2 G to T transversion, G to T transversion.